My name is Yifeng Cheng. I'm currently a postdoc in Patricia Jennings' lab. Thanks very much for Plexon's invitation, and I'm so delighted to be here today to share my research project investigating the effect of chronic alcohol exposure on behavior flexibility in rats. So alcohol, it's something that we can easily access. So I think I do not need to give a lot of introduction on that. But my main focus is to see how chronic alcohol can lead into a long-lasting effects on behavior flexibility. And of course, I'm very interested to see what's the neuron computation underlying these potential deficits. So in this presentation, I aim to provide evidence for chronic alcohol exposure induces persistent decision deficits with a newly designed reversal learning task. Also, I hope to present findings on how chronic alcohol exposure alters striatal neuron activity during decision making in this reversal learning task.
Yifeng, can I interrupt you for just 1 second?
Yeah.
I just wanted to let you know that your slides are slightly small. I don't know if it's the way that you shared the screen. It might be on presenter mode, but if there's a way to change how you shared it, it might make it where people can see the slides a little bit better.
Oh, okay. Let me try a different way.
It looks like
Sorry for the.
No, that's okay. Gabe mentions that it might be the portrait ratio. I don't know if you want to share any more about that, Gabe, or not.
Okay, let me give it a try. Yeah, maybe because the other screen I shared is a bit small, but okay, let me try. What about now?
That's much better.
Oh, okay, great.
Perfect.
All right. Yeah. So yeah, this is my experimental timeline. Due to the limited time, so I will only emphasize 2 points here. So first of all, I did a 4-week ethanol vapor exposure to just mimic the human ethanol dependence level. And all behavior and electrophysiology results were obtained after 10 weeks of the withdrawal from last ethanol session, which is beyond all the withdrawal period in the currently the field that has been used. So now I want to introduce the trial structure for this new reversal task called dynamic probabilistic reversal learning task. So before training, animals were water restricted for 24 hours to motivate them to perform the task to earn 10% sucrose reward. And each trial began with a nose poke to the sensor magazine. Then 2 levers were inserted. The animal had to make a choice left and right, and then the outcome was reported after a short delay. The outcome was indicated by 2 auditory cues. One indicates a successful reward delivered, and while the other indicates a reward omission. Then animal had to wait for a 2.5 seconds intertrial interval before they can initiate another trial. So in this task, the outcome were provided in a probabilistic way. If you plot the reward probability given choice on a 2D space, it will look like this. And the y-axis represents the reward probability for selecting the left lever, and accordingly, the x-axis is for the right lever. In this DynaPRL task, there are 3 types of block. So I label them 3 to 1 in this way. So, and you can tell block 3 has the highest contrast between left and the right lever, and the block 2 has the middle contrast, and block 1 has the lowest contrast. And of course, the challenging for animal to figure out which one, either the left or right one, it's a better option, will be different, right? Because so 3 has the lowest challenging, and of course the middle, the block 1 has the highest challenging. And we can categorize all possible block transition into easy to hard transition, or hard to easy transition, or same to same. So, and then when we look at the their choice probability on pre-reversal preferred options based on the different type of block transitions, we found that ethanol male actually showed a slower reversal behavior when they travel from easy to a hard condition. However, this effect was not been seen in the female rats. So in the following presentation, I will mainly focus on the male rats as we don't see any group differences in the female. To investigate the cognitive processes underlying their adaptive behavior in response to changing reward contingencies, I collaborated with Angela and applied the reinforcement learning model to capture trial and error learning processes. Our best fitting model was a Q-learning based model that updated the chosen action value. In this case, will be left side through a reward prediction error term and using two distinct learning rates. Condition on and of course the model can also forgetting the unchosen value with also two forgetting rate condition on whether it's rewarded or not. And for the next trial, the model will select the action throughout the softmax function with an inverse temperature termed as a beta here. And here, what we found is that CIE animal, the ethanol animal, had a higher learning rates to increment their action value when rewarded, and also had a higher forgetting rates to discount an unchosen value when not rewarded. These results suggest that chronic alcohol exposure may increase their sensitivity to the reward, causing animal to overestimate the chosen action. And meanwhile, reduced sensitivity to no reward outcome cause animal to underestimate the unchosen action. And this in turn equals to overestimate chosen action. So overall, this may lead more behavior preservation and a rigid and maladaptive pattern of the behavior in response to changing the reward contingency. So of course, next I want to understand how the striatal neuron, active or what's their dynamic in throughout the trial. And in this presentation, I will mainly focus on the outcome evaluation period. So to record a striatal activity, I just simply lowering 16 single electrodes into the dorsal medial striatum, and the single unit spike activity were recorded with Plexon system and sorted with the offline sorter. So we first align neurons' spike activities on to the onside of the outcome cues, and of course, it's not surprised that we can record a neuron that firing differently to reward and omissions here. But we also found other neurons that may respond to choice and choice outcome interactions and the value of chosen actions. So in this case, I applied the multivariate linear regression model to figure out impact of all these manifest or latent variables to see how that affect their striatal firing rates here. So many interesting results has been found here, but I will select the most two important here. Based on the coefficient, we found there is a significantly higher proportion of the neuron from the air rats that modulated by choice, and of course the modulation strength is also higher than ethanol. But when we look at the outcome here, it's the opposite. We found a significantly higher proportion of the animal from neurons from ethanol animals compared to air that modulated by outcome. And of course, the modulation strength it's higher in ethanol neurons compared to air neurons. So this actually just means that ethanol overall reduced the encoding of the choice signals in the ethanol animals, and of course enhance or overestimate outcome signals, which is similar of what we found from the reinforcement learning model. Okay, so taken together, I hope in this presentation you can find that chronic ethanol exposure impairs the reversal behavior when animals are learning in a high uncertain environment, or I will say a hotter environment. And the pre-ethanol exposure selectively enhances value updating on chosen action when rewarded and facilitates value discounting on unchosen action when there's no reward. And of course, lastly, I hope to convince you that the pre-ethanol exposure alters the neuron processing of chosen action and outcome in the dorsal medial stratum. Overall, with the modified reversal task, this study present evidence supporting chronic alcohol exposure can induce a persistent decision deficits after even 10 weeks of the withdrawal, and also this study revealed an underlying neural mechanism. In the end, I hope to give a thanks to the whole lab, especially my mentor Patricia Jenik and my collaborator Angela and Linda, and of course all the funding resources. I'm happy to take any questions in the end. And thank you very much for your listening.